Up next, we're going to hear from Nsoh Anabire from the West African Center of Cell Biology of Infectious Pathogens. So whenever you're ready, Nsoh.
Uh, can you hear me?
Yes, we're good.
All right, okay. Um, Thank you to the organizers of this conference for inviting me to give a talk. Like he has beautifully introduced me, I'm Nsoh Anabire from the West African Center for Cell Biology of Infectious Pathogens, University of Ghana. So I'll be sharing with you some parts of my PhD findings that we try to investigate why, when pregnant women, when they carry falciparum infection into pregnancy and at the point of delivery, they just rapidly lose this parasites without any therapeutical intervention. All right. So what we do know is that critical to disease phenotypes associated with falciparum infections are VAR genes and the PEF-EMP1 proteins they encode. So there are about 60 of these VAR genes that encode for different PEF-EMP1 proteins. So the VAR gene repertoire can be grouped into 5. So from groups A to groups E. Now this grouping is based on the positions of the genes on the chromosomes, the parasite chromosomes, or certain sequences on the upstream sequence positions of these genes. Now, these different groups of VAR genes have been associated with different phenotypes of falciparum disease manifestations. So the transcription of genes that encode for PFEMP1 molecules that usually belongs to group A and B usually lead to symptomatic and severe disease, usually in children. And then the VAR group C usually would transcription of these genes usually associated with asymptomatic infections. Now the VAR group D, which is also VAR1 CSA, has been found to be constitutively transcribed null isolate. But in the context of postpartum clearance of this parasite, the interest is VAR2 CSA, which is a parasite-specific antigen that is known to enable the sequestration of parasites in the placenta by binding to a host receptor, which is somehow known to be only found in the placenta, that's oncofetal chondroitin sulfate A. Now, the transcription of VAR2-CSA and ultimately expression of the protein on the surface of infected erythrocytes enables the accumulation of parasites in the placenta, leading to placental malaria, which has dire effects on both the mother and also the neonates. So what has been observed and reported amongst African women by 2 independent studies has shown that pregnant women prior to delivery, when they have these parasites, they carry this parasite into delivery. And then few hours after delivery, they just rapidly lose this parasite without any therapeutical intervention. Now, the earlier study by Nguyen, 1988, then VAR2-CSA was not discovered. By 2011, VAR2-CSA was discovered in 2003 by scientists at the Center for Medical Parasitology, which is known to be the antigen that mediates placenta malaria. So the working hypothesis was that because we know that studies have shown that in pregnancy, usually from the 3rd trimester till when the woman delivers, parasites that circulate, most of these parasites are parasites that express VAR2-CSA. Nonetheless, there are other parasites that express other VAR genes that are not VAR2-CSA. So those ones represent parasites that will cause malaria out of pregnancy, but the proportion of those parasites are usually very little, but majority of those parasites should be VAR2-CSA expressing. So the hypothesis was that prior to delivery, a higher proportion of the parasites that circulate in these women should be VAR2-CSA parasites that will transcribe VAR2-CSA and ultimately express the antigen on the surface of infected erythrocytes. Then few hours after delivery, then these parasites, since the placenta has been removed, they will lack a sequestration focus. That means that they would succumb to splenic clearance, and then such parasites will be cleared. So the proportion of parasites that would be expressing VAR2-CSA would be very, very small. So what we did was to investigate in pregnant women, and we did this in the central region of Ghana in villages called Mankesim Salt Pond. So we recruited pregnant women near delivery. So at delivery, we screened them to see if they have a falciparum infection. Then once they have falciparum infection, then we follow them in a short-term longitudinal manner between 8 to 16 hours, 24 to 48 hours, and 48 to 72 hours. So all the time point samples, perform microscopy to quantify parastemia, then PCR to quantify parastemia. Then what we did was to profile the virgin transcript profiles. So the screening data within the period that we screened, we were able to screen up about 377 women, and we used a combination of methods to screen. By rapid diagnostic screening, there were 24, and by microscopy, 17 were positive, and then by PCR, 50. But based on PCR positivity and microscopy positivity, we followed 17 women in the longitudinal period. So what we did to quantify the virgin transcription, we used a set of primers that were previously reported by our team at the Center for Medical Parasitology. So basically, the VAR genes were those primers spanning from VAR1 through to VAR3 and VAR groups A, B, and C, and BC. And the endogenous control to normalizing the transcript abundance was the fructose bisphosphate aldolase gene. And so for the cutoff for measuring whether there was a transcription of a gene or not was CT values are greater than 30. And basically the transcript abundance was calculated relative to normalizing that to the endogenous control and taking 2 raised to the power of that value. So just to reiterate or bring evidence as previous studies that have shown that these parasites by 24 hours post-delivery, they are cleared. What we did was to normalize the time point parastemia to the pre-delivery parastemia. So in the pre-delivery parastemia was set to 100%. Then we look at the percentage decrease in parastemia within the postpartum period. And as you can see, by 24 hours postpartum, then parastemia has declined to about 99%. And in most of the donors, by 48 to 72 hours postpartum, most of them were negative by PCR, and most of them just carried very low levels of parastemias. And this was confirmed by both PCR and then microscopy. So the next we did was to look at the whole virgin repertoire, the transcript abundance from pre-delivery to the post-delivery period. And the color codes just shows the individual VAR groups that are shown in the legend below. So pre-delivery, like our hypothesis, it shows that majority of the parasites were VAR2CAC expressing parasites. And if you look through to the post-delivery period, within 8 to 16 hours, you see that the abundance of parasites that transcribed VAR2CAC had reduced. And if you look at that till like 24 to 48 hours, fewer parasites transcribed VAR2CAC. So what basically this says is that within the postpartum period, parasites that transcribe VAR2CAC and ultimately would express VAR2CAC on the surface of the infected erythrocytes would be cleared because a placenta sequestration focus has been lost. However, the other parasites that will linger around are parasites that will express other VAR genes other than VAR2CAC. But what we do know is that there's substantial evidence to show that adults that have grown from childhood through to adulthood in malaria endemic area acquire PFPMP1 specific antibody responses that helps in mediating and clearing parasites that are non-VAR2CAC expressing. But we do know that VAR2CAC, immunity to VAR2CAC expressing parasites is acquired only when a person becomes pregnant. So these findings together shows that the rapid spontaneous clearance of these parasites is related to the absence of a placenta sequestration focus in the postpartum period, and also underlines the important roles of VAR2CAC specific antibody responses acquired through pregnancy, which is not acquired when an individual has never become pregnant. So to all put together, we've been able to show somewhat mechanistic role as to how these parasites are cleared in the postpartum period. So I would thank my supervisory team, that's Professor Lars Ved, who is in the Center for Medical Parasitology, University of Copenhagen, together with Mike and Gordon in University of Ghana and Pila, University of Copenhagen. The project was sponsored by the Danida, and together with the collaboration with WACCBIP in the University of Ghana in Uguchi Memorial Institute. I worked with a number of research field team members and other health personnel in Ghana, and I thank all other individuals that contributed to this study. Thank you so much for your audience. I'll be ready for any questions.
You have time for a few questions? Do we have any online questions? Dr. Augury?
Could you say a word about the glycosylation of these membrane proteins? Is there anything peculiar?
Yes, I'm not sure I heard that question very well.
Yeah.
Yeah, if you could say anything about the glycosylation of the proteins you've been using.
So actually, what we did, because of limitations to what we have in the field, this was more like a field-based project. We didn't look at the binding phenotypes of these parasites. So what we looked at was at the transcript level.
Thank you.
Yes.
Hi, my question is if you have the resolution with the gene expression data to look at expression of certain P53 and P1 domains.
So actually, yes, but the pair of primers that we used was primers that target different VAR groups, representing VAR groups that are associated with disease phenotypes. There are other set of primers that target different domains, which we didn't use for this study. So if we had used those set of primers, our team in CMP also have those set of primers that would have addressed your question.
All right. Thank you so much. So that was very interesting.
Yeah, all right. Thank you so much to the organizers as well.